Lesson 5, Gene Therapy. In this lesson, we have the topic learning objectives. These are: first is describe gene therapy and its various forms. Second is assess the issue's potential benefits and detriments to global health. So, let's move on. What is gene therapy? It is an experimental procedure aimed at replacing, manipulating, or supplementing non-functional or malfunctioning genes. So genes are specific sequences of bases that encode instructions on how to make proteins. That means gene therapy is simply the technique that modifies a person's genes to treat or cure disease. It is a medical field which focuses on the genetic modification of cells to produce a therapeutic effect. There are two types of gene therapy. First is the somatic gene therapy. It involves introducing a good gene into targeted cells with the end result of treating the patient. But take note, somatic gene therapy is not the patient's future children because these genes do not get passed along to offspring. This gene therapy involves the obtaining blood cells from a person with a genetic disease and then introducing a normal gene into the defective cell. The second is the germline gene therapy. This entails injecting foreign genes into fertilized eggs or in sperms producing cells, which will then pass any genetic changes to future generations as well. It takes place into the reproductive cell. It involves the genetic modification of germ cells that will pass the change on to the next generation.
Methods of gene therapy. There are 7 methods of gene therapy: the physical, retrovirus vectors, other viral vectors, targeted gene transfer via receptors, artificial chromosomes, site-directed recombination, and activation of genes-related function. Under physical method, there are direct injection, liposome-mediated DNA transfer, and electroporation. In direct injection, DNA can be parenterally, which could be considered for Duchenne muscular dystrophy or DMD. It is an alternative approach, uses particle bombardment or gene gun technique in which DNA is coated onto mental microparticles and fired from a ballistic gun on cells or tissues. This technique is used to transfer the foreign DNA and its transient expression in mammalian cells in vitro and in vivo as well. It can be cross the physical barriers like skin, muscle layer, for which it is used for vaccination. In electroporation, the external electric field is applied to the protoplast, which changes the electrical conductivity and the permeability of cell membrane are taken up to either cytoplasm or into the nucleus. Electroporation is well suited for the transformation of plant cell and protoplast. Extensive work has been carried out regarding transformation of cereals using protoplast. Both linear and circular DNA can be transformed into the plant tissue. Intact plant cells of monocots have been transformed by electroporation. During electroporation, protoplast or intact plant cells are taken in electroporation chamber fitted with parallel steel electrodes. Third one is liposome-mediated DNA transfer. Spherical vesicles, which are made of synthetic lipid bilayers, which imitate the structure of biological membranes. DNA to be transferred is packaged into the liposome in vitro and transferred to the targeted tissues. The second method of gene therapy is retrovirus vectors. These are RNA viruses which possess a reverse transcriptase activity, enabling them to synthesize a complementary DNA. Tumor retroviruses is widely used for generation of recombinant virus. These are produced at low titers as all the viral genes are deleted. Example of tumor retrovirus is Maloney's murine leukemia virus. Maloney's murine leukemia virus are retroviruses named for their ability to cause cancer in murine hosts. Oncoretroviruses. The cells that divide shortly after infection can only be transduced by oncoretrovirus. The pre-initiation complex is excluded and their entry is restricted into the nucleus as they can only enter when nuclear membrane dissolves during cell division. The target cell for this viral vector is limited. Recombinant lentiviruses. These are being developed that are non-pathogenic to humans and have the ability to transduce stationary cells. The third method of gene therapy is the other viral vectors. These includes herpes simplex virus vectors and baculoviruses. Herpes simplex virus vectors is a 150 kb double-stranded DNA virus with a broad host range that can infect both dividing and non-dividing cells. The insert size is comparatively large, 720 kg, but have a disadvantage short-term expression due to its inability to integrate into the host chromosome. Baculovirus. They can take up very large genes and express them highly efficiently. They help in recombinant protein expression in insect cell. They can infect hepatocytes as an only mammal cell type and the gene expresses under the control of either mammalian or viral promoter. The fourth method of gene therapy is targeted transfer via receptors. It can be both viral and non-viral mediated gene transfer. Viral vectors attach to the surface receptors through viral surface components and internalized. In non-viral mode of receptor-mediated endocytosis, DNA is first coupled to a ligand that binds specifically to a cell surface receptors and causes transfer of the DNA into cells by endocytosis. Coupling is done by linking the receptor molecule with polylysin, followed by reversible binding of the negatively charged DNA to the positively charged polylysin component. The fifth method of gene therapy is artificial chromosome. These are cloning vectors that can carry DNA insert orders of magnitude larger than is possible with plasmids. Like other cloning vector, artificial chromosomes contains DNA sequences elements that are necessary for replication and stability of the molecule in their host cell and for its partitioning to daughter cells upon cell division. Another method of gene therapy is site-directed recombination. The homologous recombination is the natural recombination phenomenon by which nucleotide sequences are exchanged between two chromatids in cell division. This natural ability of the cell is utilized to shuffle the target construct to the genomic DNA. And the last method of gene therapy is the activation of gene-related function. Gene delivery and activation. The prerequisite of gene therapy is the delivery of vectors to the target cells at the target area of tissue. The gene should be in higher concentration at the site of target cells so that the cells can easily take up the corrected or wild gene. When the gene reaches its destination, it has to be activated to produce the desired protein encoded by that specific gene. However, such gene delivery and activation are the biggest obstacles faced by the gene therapy in
Target cells for gene therapy. This is a visual representation of gene transfer. Peripheral blood lymphocytes. Genetically corrected T cells are currently under investigation as a treatment for severe combined immunodeficiency disease resulting from a lack of ADA. Monthly injections of these ADA-corrected T cells have resulted in measurable ADA activity in the peripheral blood and in the vivo production of antibody to blood group antigen. Keratinocytes. Keratinocyte gene therapy. The gene that will correct the disease by expressing a normal protein or enzyme is inserted and expressed in keratinocytes. Keratinocytes have significant potential as a target cell of gene therapy in the treatment of both systemic diseases as well as skin diseases caused by the genetic defect in keratinocytes. Hepatocytes. Hepatocyte-based gene therapy may be used to replace a missing gene product, confer proliferating ability to cultured hepatocytes, prevent allograft rejection, massively repopulate the host liver, or grow xenogenic hepatocytes in mammalian liver. Gene transfer into isolated hepatocytes can be accomplished via non-viral or viral vectors, the viral vectors being more useful this time. Fibroblast. Dystrophic epidermolysis bullosa or DEB is due to mutations in the type 7 collagen or C7 gene. Potential therapies for DEB include ex vivo gene therapy and intradermal injection of gene-corrected DEB fibroblasts. Lentiviral vectors expressing C7 or recombinant C7 itself. With regard to molecular engineering, the dermal fibroblast has advantages over epidermal keratinocytes for delivering C7 to DEB patients. Hematopoietic stem cells. Hematopoietic stem and progenitor cells can be an alternative to allogeneic transplantation in the treatment of primary immunodeficiencies. Early trials also highlighted the risk of insertional mutagenesis and oncogene transactivation associated with the first generation of gamma-retroviral vectors. Vascular endothelial cells. Abel et al. Designed lentiviral vectors or LVs enabling specific gene delivery into endothelial cells in vivo. This opens new perspectives for gene therapy of hereditary disorders, cardiovascular diseases, and cancer. Airway epithelial cells. The airway epithelium is pseudostratified in the large airways, becoming columnar and cuboidal in the small airways. The major cell types are ciliated, columnar, undifferentiated, secretory, and basal cells. Tumor cells. Gene transfer is a new treatment modality that introduces new genes into a cancer cell or the surrounding tissue to cause cell death or to slow the growth of the cancer. This treatment technique is very flexible, and a wide range of genes and vectors are being used. Skeletal muscle myoblast. Myoblast transfer therapy and gene therapy have both been proposed as potential treatments for inherited myopathies, such as Duchenne muscular dystrophy or DMD. The success of myoblast implantation in mouse models, where problems such as immune rejection are easily overcome, have led to similar experiments being attempted in Duchenne patients with limited, if any, success. Gene therapy is a promising technique to treat or prevent complex conditions. A corrected version of gene is injected directly into your body, thereby eliminating many of the complications that occur with more standard therapies. While gene therapy is still considered experimental for many conditions, early outcomes are encouraging.
Continuation of the gene therapy. So, these are some advantages of gene therapy. Gene therapy can cure genetic diseases by addition of gene or by removal of gene or by replacing of mutated gene with connected gene. The second is gene therapy can be used for cancer treatment to kill the cancerous cells. And the third is therapeutic protein is continuously produced inside the body, which also reduces the cost of treatment in the long term. So the advantages of the gene therapy is that it provides an opportunity for the affected individuals to lead a life to a normal, Some gene therapies may even offer the chance to survive because the genetic disorder is diagnosed as being terminal without an intervention. And then, may ada po gihap disadvantages and gene therapy. So, the gene therapy is very expensive and will never be cost effective enough to merit high social priority. Germline gene therapy would violate the rights of subsequent generations to inherit a genetic endowment that has not been intentionally modified. And gene therapy would open the door to attempts at altering human traits not associated with disease, which could exacerbate problems of the social discrimination. So the disadvantage of the gene therapy, for example, and medical procedure, sometimes diri na ini na work that's very expensive pa. Gene therapy does provide means of treating diseases, but still this therapy is not very common because it has some limitation. Maybe the therapeutic genes which are inserted in the body not accepted by the immune system and the viral vectors are not useful because they can trigger their pathogenic features in the body.
Advantages of gene therapy. There are 5 advantages of gene therapy. The first one is germs-led gene therapy offer a true cure and not a simply palliative or symptomatic treatment. That means the gene therapy are always offer the trusted and tested cure to the patient because that will make the patient survive. The second one is germs-led gene therapy may be the only effective way of addressing some genetic disease. Gene therapy are the only one that can make the genetic disease effectively. The third one is by preventing the transmission of disease genes, the expenses and risk of somatic cell therapy for multiple generations is avoided. Somatic cell therapy or somatic gene therapy can make so many generations, but we should know how to avoid it because we don't know what will happen to our body, to our children, and to the next generation.
Disadvantages of gene therapy. Germline gene therapy experiments would involve too much scientific uncertainty and clinical risk, and so the long-term effects of such therapy are unknown. Such gene therapy would open the door to attempts at altering human traits not associated with disease, so which could exacerbate problems of social discrimination. As germline, gene therapy involves research on early embryos and affects their offspring. So such research essentially creates generations of unconsenting research subjects. Gene therapy is very expensive and will never be cost effective enough to merit high social priority. Germline gene therapy would violate the rights of subsequent generations to inherit a genetic endowment that has not been intentionally modified.